my PhD research, I'm studying human differences of sex development. These are a group of conditions where the reproductive anatomy develops atypically. So these occur around once in every 4,000 births. One example are cases where the testes don't develop as they normally would, meaning that they don't produce testosterone. So even though a baby might be genetically male with XY sex chromosomes, they actually develop externally as female. This is caused by damaging variants in the genes that control how the testes develop. A few years ago, just 13% of these patients received a genetic diagnosis. With our group's research, we've increased this diagnostic rate to 45%. We've done this using next-generation sequencing. This is where we look at all of the genes in our patients and try to identify the specific variant that might be causing their condition. But to get this diagnostic rate higher, we need a way to prove that these new patient variants are actually disrupting testis development. Traditionally, mouse mutant models have been used to study these new genes suspected of causing differences of sex development. However, in many cases, these animal models don't replicate human differences of sex development, limiting the translation of these findings to patient diagnosis. The aim of my PhD research is to create a human-specific model of developing testes using induced pluripotent stem cells. Induced pluripotent stem cells, or iPS cells, have the ability to become any human cell type. The type of cell they choose to become depends on their environment. So much of my work till now has been focused on trying to figure out what cues I need to give to these cells so that they choose to become testis cells. By adding certain growth factors to the stem cells, we can see that testis genes are being switched on. So already we're getting cells really similar to what you'd find in the testes of a human embryo. What's more is that these cells are even organising themselves into testis-like structures in a dish. The IFER fellowship will help me to build on this preliminary work through two main aims. The first is to make this protocol xeno-free by replacing the animal-derived products that we currently use in our protocol. This will involve acquiring these new reagents and re-optimizing the stem cell to testis differentiation protocol with these animal-free alternatives. The second aim is to replace the use of animal-derived antibodies. In our lab, we've generated iPS cells that fluoresce when testis-specific genes are turned on. With the IFER fellowship funding, I will optimize the use of these iPS cells in our testis differentiation protocol, and this will allow us to monitor gene expression in differentiating cells without the need to use animal-derived antibodies. Developing this protocol would remove the dependence on animal models when studying differences of sex development, as well as improve patient diagnosis and clinical management. I'm very grateful to receive the International Foundation for Ethical Research Graduate Fellowship, and I really look forward to contributing to the development of non-animal models.